Awesome, thank you for that, Matt. So now I'll talk a bit more about surface registration and cortical parcellation as tools for investigating the organization of the cerebral cortex. I don't have any disclosures. So just to give a brief overview of what I want to talk about today, first is how we model the cerebral cortex as a surface, kind of building on what Matt has already talked about. Then surface-based registration, a little bit more of the technicalities involved with that, and then cortical parcellation and why we might use that as a tool. So why model the cerebral cortex as a surface? So if we think about the organization of the cerebral cortex, we know that it's the outermost layer of the cerebral hemisphere is made up of about 16 billion different neurons. And when we're thinking about the organization and the development of the cortex, the cells, the neurons, they start from the subventricular zone and they migrate outwards in an inside-out fashion towards the outside of the cerebral cortex. With each of, and they form these cortical columns that we might be aware of. The function of these columns are determined by what is known as morphogen gradients. So these are kind of patterning centers in the brain that tell the brain if it's anterior or posterior, medial or lateral. The major consequence of this organization is shown on the right-hand side of the screen, where we're looking at how the function of the cortex changes as we move across its surface. In particular, these are electrophysiological experiments by Hubel and Wiesel looking at the orientation selectivity of neurons in the visual cortex, or the primary visual cortex. And what they found was, with this oblique arrow, as they passed their electrode tangential to the cortical surface, what we call the orientation selectivity of these neurons in area V1, they rotated. So you can see these initially horizontal line rotating all about 180 degrees. Alternatively, if we look at the arrow that is vertical, what that demonstrates is that no matter how far you put the electrode through the cortex, all of those neurons in that single column share the same function. So to summarize all of this, basically the functions of the cerebral cortex are encoded as distance tangential to the surface. Jumping tack a bit, the whole point of neuroimaging is to find differences between individuals. And so in this study here, what we're looking at is preterm and term-born neonates, and we're looking at differences in the functional connectivity of their primary sensory networks. And this is a nice study by Ear et al. Unfortunately, when we acquire data, we can't assume that each voxel represents the same anatomical point across individuals because of differences in head position, brain size, and shape. So what I've done here is I've placed my cursor at the same voxel coordinate, XYZ position, in both brains, and you can see that it's representing a different anatomical position across both of them. And so what we do to bring all of these individuals into a common coordinate space is image registration. We take one brain and we try and match it to a template. And so on the left-hand side of the screen, you can see we just have an individual subject, and it's in its template, or it's in its own or native space. We then have the same brain in the middle, and it's been registered to a template. And then on the rightmost image is the template that we're trying to align everything to. And we're simply doing this based on differences in intensity. So we're trying to get grey matter to align to grey matter and white matter to align to white matter. If we focus on one part of the cortex in the frontal lobe, we can see these boxes demonstrating how we deform or warp the cortex to match it to the template. What you might also notice is that the cortex in the template, the cerebral cortex, is very, very blurry compared to an individual subject, especially in areas where there's high folding variability, so in the frontal and parietal lobes, for instance. And this is a result of volumetric image registration not being able to completely overlap the individual cortical ribbons of all subjects. So this is a nice paper by Tim and Matt, and what this figure is showing is different parasagittal slices of the cortex, and for 210 participants, individual cortical ribbons overlapped, where areas of red represent areas of high overlap, and darker colors representing areas of lower overlap. And so what you can see is that the histogram on the right demonstrates that for a significant proportion of voxels that have overlapped, for a significant proportion of voxels in individual cortical ribbons, they're not being compared with cortex, they're being compared with white matter or CSF. So this makes it really difficult to try and compare like with like, in this case cerebral cortex with cerebral cortex. And so the entire point of modeling the cerebral cortex is to inflate our very highly folded cerebral cortex into a sphere. So this cow, we're just blowing it up like a balloon until it represents a sphere. We're doing the exact same thing with the cerebral cortex. As an aside, and as Matt has already mentioned, it makes it much easier to appreciate the cerebral cortex in its entirety rather than having to scroll through anatomical slices. And so what we're interested in surface-based analysis is what we call this geodesic distance. And so what I've got here is a zoomed up version of the central sulcus, where we have area 4, the primary motor cortex, represented anteriorly or upwards, and then we have the sensory cortex, area 3B, represented posteriorly or below in this figure. And what the orange line represents is the distance between the neurons in these different areas, the actual difference if we were to take the brain and unfold it. What we can see is that this difference is, this orange line is much, much, much larger than the red line, which represents the distance between these two areas that would be encoded in a volumetric representation of the brain. And this is, Matt's already alluded to, this is what happens when we don't or aren't able to account for the geodesic distance between different cortical areas. So in the middle we have a group average surface and probabilistic cortical areas across the entire cortex, with red representing higher overlap across individuals. And these blue arrows point to the corresponding volumetric representation. And so we can see that there's a very large drop in the overlap of these areas across the cortex, not just for area MT, but also in the sensory cortex, area 55B, that language area that Matt was talking about, and the frontal lobe as well with area 46. So it makes it much more difficult to compare like with like when we're using volumetric representations. So now, hopefully I've kind of motivated why we care about doing surface representation. How do we then take our surface representations and put it into a common coordinate system like we would for volumes? So the first thing I want to start off with is imagining what a volume-based representation of the cortex would look like versus a surface-based representation. So we can see that the smallest element of a volumetric image is known as a voxel, and so if we're looking at a patch of cortex, it's made up of many, many voxels, and we can calculate the volume of that area. And depending on how big or small the voxels are, we may be able to very accurately delineate the cortex, or what is more likely is that there will be some voxels that have captured both grey and white matter, and we get this effect called partial voluming. The idea with surface-based analysis is to get what we call subvoxel resolution of the cortex. And we're interested in two different surface two different surfaces of the cerebral cortex. So the first is this white surface, which represents the interface between the grey and the white matter. And then we get the peel surface, which is the representation between the outermost part of the cortical grey matter and the CSF. And this representation allows us to calculate a number of properties of the cortex. So we can calculate cortical thickness, and we can also calculate surface area. And as Matt has already alluded to, if we zoom in on one of these surfaces, we're no longer dealing with voxels, but these very, very small triangles where each point on a triangle is known as a vertex. And so the point of surface registration is not to match voxels to voxels, it is to try and match vertex to vertex across individuals using a property of interest. So just to kind of break down what image registration is in its simplest sense, what we're trying to figure out is how good does our individual match to the template. The second thing we want to specify is how we are allowed to warp or distort this brain so that it matches the template. In most image registration softwares, we're not doing this for every single point. We're only doing this for a subset of points. And so for those vertices that we haven't explicitly moved, how do we interpolate those to match the template? And then the last part is finding the parameters that maximize the similarity between the two brains. And so for surface registration, what we're dealing with is the sphere on the left-hand side represented by A, and we can see that it's got made up of all those small triangles. And like I said earlier, we don't want to move every single triangle or every single vertex. It's quite computationally expensive. What we can do instead is choose a subset of points to move. And so hopefully you can see in B and C, there is the very small triangles represented in grey, and that's the data that we're working with. What we do is to select a subset of points that we want to move, and that's called our control grid. And those are the red lines. And then what we want to do is choose or select a subset of points where we're allowing our control point to move. And that's called our label grid. And what we're doing is we're basically moving around our vertex to each of those individual crosses and trying to find the best match based on folding or some other property. Matt's already talked about FreeSurfer, developed by Bruce Fischel in the late 90s, early 2000s. And FreeSurfer uses different cortical folding patterns. So these are very easy to obtain simply by passing your volumetric images through FreeSurfer. What we can see on the left-hand side is the folding template, with red representing the sulci, or the dips, and the green representing the gyri, or the peaks. The top row represents individual cortical folding patterns in their native space, and then the bottom row represents them in their template space. And so we can see that most of the red falls within the dark boundaries, that is the template. But you'll notice that there are some areas where that doesn't happen. Why is this? This is because cortical folding is extremely variable from one individual to another. And image registration is what we call diffeomorphic, which means that we're not allowed to break this mesh. We have to keep individual vertices connected from one to another. We're not allowed to break it and then rematch things. And so it's unclear, for instance, if we look at type 1, for instance, it's not clear how we would match this to this. Are we going to completely eliminate this second sulcus here, and so that this becomes the template, or are we going to try and duplicate that somehow so that it matches to this? And going a little bit further with folding patterns, this is another study by Bruce Fischel that was done in 2008. And what they were interested in is looking at what the relationship between cortical folding patterns was with cortical areas defined ex vivo, so under a microscope. And so what this image is showing is the probabilistic overlap of cortical areas across the cortex when these areas are defined under a microscope. And as we expect, areas like V1 here, V2, and the part of the primary motor cortex, there's very good correspondence between a cortical fold and that area defined histologically. When we move beyond these primary areas and we move into things like area 44 and 45 into the frontal lobe, what we can see is that there isn't very good correspondence between cortical folding and cortical areas ex vivo. And so this is basically highlighting, if we're interested in cortical areas or cortical function, perhaps driving our surface registration with cortical folds is not the best idea. So if we're not going to use cortical folds, what else can we use? This is a study done by Mertzabanku in 2010 where they said, well, if we're not going to use cortical folds, why don't we use task-based fMRI? And so they drove their registration using task contrasts. And what you can see here is the results of the group average contrast when they have driven registration by folding. And if they then drive the registration with the task contrast itself, this is what they get. And so hopefully you can appreciate that there's better overlap in the fusiform face area and the parahippocampal place area when we're driving registration with measures that more closely relate to what we're interested in. But that still falls short as a tool for investigating the cerebral cortex in its entirety, because we've only been able to drive registration with a single task contrast. So that will work for these areas of interest, but for the rest of the cortex it won't provide very good overlap. So we've talked about cortical folding and task-based function. These have been single modalities that we're allowed to drive registration with. In 2014, my supervisor, Dr. Emma Robinson, developed MSM, which Matt has already talked about. The most important advance in this software is that we're now able to choose any different sort of surface feature to drive registration. It can be multiple features that we're allowed to drive registration with. And so at the top here, we've got free surfer and folding-based registration. And we're looking at task overlap in these red clusters and these blue clusters. And so what we can see is that they do a reasonable job and there's not too much difference between them. If we then drive registration with something called the T1/T2 ratio, or myelin in this case, we can see that we do a slightly better job of aligning parts in the frontal lobe here, which are the frontal and the posterior eye fields. If we then use, I think it's 26 resting state networks, so we're trying to align 26 different modalities rather than just one, we get much better overlap. And so the takeaway point of this is we can use multiple modalities that are better related to cortical areas or the measure of interest to drive registration rather than being limited to just folding. And Matt kind of alluded to it before, but this software was then used and optimized in a configuration known as MSM-all, where there were 40 different resting state networks, T1/T2 myelin maps, and some visual topic maps to drive very accurate registration between individuals. And what we can see going down is we have this MSM-all based registration configuration compared to our folding based registration configurations. and we have these probabilistic areas that I've showed you before. We get much better overlap when we use MSM-all compared to single folding-based measures. And because we're only looking at selected cortical areas, what we can do is calculate, I guess, the inverse of all probabilistic cortical areas represented on the surface and get what we call an uncertainty map. And so this is saying, how certain are we that a vertex at this point here represents one area or the remaining areas? So more black is better. So hopefully I've convinced you that surface-based registration provides far better overlap across individuals compared to volume, and that if we can, we should be using features that are better tied to cortical areas. So fMRI, task-based fMRI and resting state, or T1 over T2 myelin maps rather than folding. But if you only have folding, that does a far better job than volume. Moving on to the next part of the talk, why are we interested in parcellating the cerebral cortex? So if we go back to the fundamental organizations of the cerebral cortex, we know that it is a layered structure. In general, there are 6, and we can. There are 2 different types of properties that we're interested in when we're talking about the layering of the cerebral cortex. First is cytoarchitecture. So how are the neuronal cell bodies organized? What is their density like across the 6 different layers? The next is myeloarchitecture, and that is how the axonal fibers are oriented, how densely they're packed in the different cortical layers. So this is just a very generalized representation of these 6 different layers and their cyto and myeloarchitecture. And this layering is like quite appreciable under the microscope. What many scientists have noted is that different cortical areas will have different cytoarchitectural here and myeloarchitectural organization. And that actually when you're moving from, say, M1 to area 3A to 3B, it's the change in the cyto and myeloarchitecture is quite dramatic. And so the idea is that these areas can be defined based on their myelo and cytoarchitectural organization, and that these represent cortical areas. Some of the earliest efforts to parcellate the cortex relied on cortical folding, and most people would have represented cortical areas. Most people would have represented the cortex using this system that is distributed as part of FreeSurfer. This is very good, in that you have very well-defined anatomical regions, but unfortunately these don't capture the underlying functional and structural organization of the cortex itself. So what Thomas Yao and collaborators did is tried to parcellate the cortex based on resting state function. And so you get, I think it's 7 and 17 network representations. And these are more aligned to the kinds of features of the cortex that we're interested in. But these networks were defined by driving registration using cortical folding, and they're only based on a single feature, which is resting state function. And so what we can do is try and define a parcellation using different modalities, and the idea of a multimodal parcellation. The idea or the concepts is that registration should be driven by features that better reflect cortical organization, so that MSM-all that I was talking about before. We know that the cyto and myeloarchitectural boundaries of different cortical areas are in agreement, and so that when we're looking in vivo, there should also be agreement between, say, resting state function and T1 over T2 ratio. So that we're not over-parcellating the cortex, we should also be looking for the effect size, so how different is the cortex on one side of the boundary to the other. Ideally, they should be statistically significant with large effect size. And coming all together, this should reflect what we see at a microscopic level, so ex vivo. And so what I've shown here is an example that my lab will be familiar with, and Matt's lab as well, is this area 55B. So it's this very small area just immediately anterior to the primary motor cortex. And it's a very good example of how to define a cortical area using multiple different modalities. And so in this myelin map, what we can see is that area 55B is relatively low intracortical myelination compared to its adjacent cortical areas. We know that this area is also activated when individuals are listening to stories in a task fMRI paradigm. And if we're looking at functional connectivity across the cortex, this has quite a unique connectivity profile in that it's highly functionally correlated with this area called the perisylvian language area. But that if you place a seed in the lateral interparietal area, the frontal and the posterior eye fields light up, but area 55B is not activated at all. And so we can see very clearly that area 55B is defined across multiple modalities. So how do we do this in an observer-independent manner? Well, what we can do is calculate how fast these different features are changing over the cortical surface in what we call a gradient. And so we can see that the gradients of these different spatial features overlap very nicely, and that actually this is an area that has been defined ex vivo by Adolf Holpf in the 1950s with one of his myelin maps that he drew. And then we get to this multimodal parcellation that Matt published in 2016, where you've got 180 different cortical areas over a single hemisphere. Now, a group average cortical parcellation is good in most cases, but there will be instances where it won't align with individual anatomy very well. And so, again, if we look at area 55B, in a typical individual, it will align with the group average. But what Matt and David Van Essen noted is that in a significant proportion of individuals, they found what they call atypical topography. So actually, instead of area 55B being a single area, it's split into two different areas. And image registration, being diffeomorphic, we're not allowed to break it and reattach all the vertices together. There is no way that image registration, no matter how good it is, will capture that split topography. And again, in another subset of subjects, area 55B was pushed upwards, so we call it shifted. And if you're using a group average, where this white line is, that will incorrectly assume this area is 55B, so at an individual level, there'll be misalignments. And so parcellation is a very good tool, if you do it accurately, to get better correspondence across individuals where we think there might be topology breaking variation between individuals. Just to plug that, Matt talked about the HCP and the multimodal alignment that exists across individuals. Myself and Emma and Matt and a group at Oxford have now done this for the UK Biobank, so you can get MSM all aligned data as part of the UK Biobank. And just a quick disclaimer is that cortical areas are only one level of organization within the cortex. Sophie's very happy. And there are many other ways that we can represent the cortex. So Janine will be talking about nodes and networks. Sophie will be talking about gradients later on, so don't think that cortical areas are the only way to investigate the cortex. And that's the end of my talk.